Welcome back to the deep dive, where we take the densest molecular mechanics and really try to extract the pure distilled knowledge you need.
Today is a big one. We are undertaking a monumental task, really. We're cracking open Chapter 15, which is all about cell signaling and signal transduction.
Right.
I mean, this is basically the operating system of complex life. It's the information superhighway that defines how individual cells can cooperate to build a whole organism.
I love that the source material immediately goes to that classic John Donne quote, no man is an island.
Exactly.
And it just applies so perfectly to cells. They aren't autonomous little blobs. They're highly specialized, sure, but they absolutely must communicate and coordinate everything. Growth, metabolism, movement, all of it.
And if they stop talking, or, if they start sending and receiving the wrong messages, the whole system just falls apart.
Yeah.
So understanding signaling is, it's foundational.
Yeah.
Especially because so many devastating disorders, and cancer is chief among them, are fundamentally a loss of that communication control.
It's when a cell just decides it doesn't need to follow the rules of the neighborhood anymore.
That's it. And then catastrophe follows.
So before we really jump into the mechanics, we kind of need a mental map of the players, right?
Right.
And the chapter starts by showing us this amazing X-ray structure of the entire command center for one of these key messages.
It's such an essential conceptual anchor. What you're seeing is a representative of the largest protein superfamily we know of. It's a G Protein-Coupled Receptor, or a GPCR.
Okay.
And this receptor sits in the membrane, it receives the message, and it's complexed with its main internal relay, which is this inactive three-part heterotrimeric G protein.
So you have the external antenna, the GPCR, with those classic 7 helices that snake across the membrane.
There's 7 transmembrane helices.
And then the internal transmitter is just waiting right underneath it. That whole complex is basically designed to translate an external molecular handshake into a cascade of internal events.
Our mission today is a comprehensive step-by-step tour through these communication mechanics. We're going to follow the chapter structure precisely. The goal is to build that robust foundation you need to really understand how cells talk and, how we can modulate that conversation.
Right. Let's start with the absolute basics, the language. The book calls them extracellular messenger molecules.
Right. These are what cells use to talk to each other. And the specific molecule that binds to the receptor, that has a name, it's called the ligand.
And here's our first really deep conceptual insight, and it links right back to that island idea. The response of the cell doesn't just depend on the ligand it gets.
No. It depends on the internal context. It's all about the machinery the cell already has inside.
This is such a crucial point. It shows the control is internal. It's not just dictated by the environment. I mean, think about that beta-2 adrenergic receptor we just mentioned. If the ligand adrenaline hits that receptor in a liver cell, the internal machinery goes, okay, break down glycogen.
But if that exact same adrenaline molecule hits the exact same beta-2 receptor in, say, a smooth muscle cell lining a blood vessel,
the result is the total opposite.
The total opposite, muscle relaxation. The external stimulus is identical, but the internal programming dictates a completely different physiological outcome.
To categorize these conversations, the source material breaks it down into 3 modes of signaling, and it's all based on distance.
Right, how far the message travels. The shortest distance is autocrine signaling.
The cell talking to itself.
Exactly. The cell produces a messenger and then responds to that same messenger. It's often to, reinforce or maybe inhibit its own activity.
Next up is paracrine signaling, which I think of as like local gossip.
That's a great way to put it. The messengers only travel really short distances to cells that are right nearby. And there's a reason for that. They're often chemically unstable, or they get degraded by local enzymes really fast, or they just get stuck to the extracellular matrix. They're designed for a quick local impact.
And finally, the long-distance call, endocrine signaling.
These are your hormones. They have to be stable enough to travel all the way through the bloodstream to reach distant target cells across the entire body. It's the body's postal service.
Okay, so now let's map out the general flow of information, the actual signal transduction pathway. The chapter lays this out so clearly in 9 conceptual steps, starting outside the cell.
Right. Step 1 is just the release of the messenger. Step 2, the messenger binds to the receptor on the outer surface. And then step 3, this is the key moment. That binding causes a crucial conformational change. It's a shapeshift that relays the signal across the membrane to the inside.
And that's the moment the external signal becomes an internal instruction.
That's it. And at this point, the path can kind of split into 2 major routes. Route A is all about generating these small, mass-produced molecules.
So the signal gets relayed to a nearby effector enzyme. That's step 4.
Yep, and then that enzyme generates a small, rapidly diffusible substance, which we call the second messenger. That's step 5. And these are the famous ones, like cyclic AMP or KMP or calcium ion.
Route B is a bit different, and it's often used by another class of receptors, the receptor tyrosine kinases. Here, the cytoplasmic part of the receptor itself becomes like a recruiting station.
A recruiting station, I like that. It's a platform where all these specialized intracellular signaling proteins can dock and interact. They use these little modular domains, like one called an SH3 domain.
So regardless of the route, the process then moves deeper into the cell. Step 6 is activating a protein at the very top of an intracellular signaling pathway,
Which kicks off step 7, this sequential operation of a whole complex pathway. You have to think of it like a relay race. And these signaling proteins are highly modular, so they're built with standardized parts that let them link up with all sorts of different partners.
And that relay continues until the signal finally reaches the target proteins in step 8.
Which leads to the final culmination, the cellular response, which is step 9. And that action can be anything. Turning a gene on or off, changing metabolism, starting cell movement, or even telling the cell to die.
So what's actually facilitating all these switches, these on-off changes in protein function?
Overwhelmingly, it's phosphorylation. The cell's molecular switch mechanism is built on just adding or removing a simple phosphate group.
The scale of this is it's staggering. The source material notes there are over 500 different protein kinases that add the phosphate groups. They're the on switch.
And about 150 different protein phosphatases that remove them. They're the off switch.
And what's really profound here, what you have to get, is that phosphorylation isn't just an on-off switch.
No, it's a mechanism that totally changes a protein's behavior.
It
can
can
activate or inactivate an enzyme, sure, but it can also fundamentally change what other proteins it can interact with.
It can even make it physically move between cellular compartments, like from the cytoplasm into the nucleus.
Or it can flag it for complete degradation. It's the central binary switch of the cell.
And we can see the immediate relevance when that balance is lost. The chapter includes this really powerful visual example. It's figure 15.4, comparing the frequency of tyrosine phosphorylation in different breast cancer cells.
And the detail that's so striking is that triple-negative breast cancer cells, which are notoriously aggressive,
Right.
they showed markedly higher levels of overall tyrosine phosphorylation compared to the other types. It's just lit up.
This excessive signaling, this persistent on state, is thought to correlate with the loss of a specific phosphatase, a protein called PTPN12.
So if the enzyme that's supposed to remove the activating phosphate is missing,
The proliferative signal just stays locked on, driving uncontrolled growth,
it's a perfect example that shows the off switch is every bit as important as the on switch.
which brings us to termination.
Yeah,
You have to be able to stop these signals.
absolutely. Beyond phosphatases, you can just have enzymes in the extracellular space that destroy the original messenger. Or the cell can pull the receptor itself inside, a process called internalization, and then degrade it. That temporarily makes the cell less sensitive.
Okay, before we dive into the two major receptor types, let's just quickly acknowledge the immense diversity of the messengers themselves.
They're so chemically diverse. They range from tiny amino acid derivatives like neurotransmitters,
To even gases like nitric oxide, NO, and carbon monoxide, CO.
and you have lipid-soluble steroids from cholesterol, and these complex fatty acid derivatives called eicosanoids, which are really important for local inflammation, pain, and blood clotting.
And of course, thousands of polypeptides and proteins that act as hormones or growth factors. It's an incredibly rich language.
And to manage all that diversity, cells really rely on 4 major classes of receptors to mediate the signal transduction.
First up, the massive family we already introduced. The G Protein-Coupled Receptors, or GPCRs. They've got the 7 transmembrane helices, and they relay their signal through those G proteins.
Second, Receptor Protein-Tyrosine Kinases, the RTKs. These usually have just a single transmembrane helix. They typically bind growth factors, they dimerize, and then they activate their own internal kinase domain to phosphorylate tyrosines.
Third, ligand-gated channels. These are pretty simple. The receptor is an ion channel, and the ligand binding directly opens or closes it.
Right, super fast. Neurotransmitters use these a lot to generate nerve impulses in milliseconds.
And fourth, steroid hormone receptors. Since steroids are lipids, they can just diffuse right across the membrane.
So their receptors are inside the cell, they're intracellular, and they basically function as ligand-regulated transcription factors that go into the nucleus and control genes.
So for the rest of this deep dive, we're going to focus really intensely on those two major membrane players, the GPCRs and the RTKs. They just dominate how cells respond to most external signals.
All right, let's start with the GPCRs, also known as 7T-M receptors. I mean, this is the largest protein superfamily in animal genomes.
And they govern everything from hormone responses, like our adrenaline example, to the super subtle nuances of smell and vision. Table 15.1 just gives a tiny glimpse of their ligands, everything from light and acetylcholine to opioids.
The structure is very standardized. 7 helices crossing the membrane, a binding pocket on the outside, and a cytoplasmic tail that talks to the G protein on the inside.
The activation mechanism is so elegant, the chapter uses the example of rhodopsin, which is the GPCR for vision.
Right. So a photon of light hits this little molecular cofactor called retinal, and it physically flips its shape. It isomerizes.
That one little flip disrupts a tiny ionic bond that was holding the receptor in its inactive state.
And that destabilization causes one of the transmembrane helices, helix number 6, to physically tilt and rotate outward. It acts like a lever that opens up the G protein binding site on the inside of the cell.
So this newly activated receptor can now interact with the inactive 3-part heterotrimeric G protein made of the G-alpha, G-beta, and G-gamma subunits,
which initiates the core G protein cycle, which is laid out in figure 15.6. Step 1, the activated receptor binds the G protein, which has a GDP molecule stuck to its G-alpha subunit.
And step 2, that interaction induces this massive conformational change in G-alpha.
And this causes G-alpha to let go of its GDP and immediately kick up a higher energy GTP molecule. This GDP-GTP exchange, this is a major point of signal amplification. One receptor can activate hundreds of G proteins while it's active.
Once that GTP is bound, the relay is complete. That's step 3. The G-alpha GTP subunit physically breaks away from the G-beta-gamma complex.
And here's a key point. Both the separated G-alpha GTP and the free G-beta-gamma complex are now active signaling molecules. They can float off and activate or inhibit different effector proteins.
But here is the central control mechanism. G-alpha is inherently unstable. It has its own built-in GTPase activity.
It's a molecular timer. In step 5, it slowly hydrolyzes its bound GPP back to GDP.
And that hydrolysis is the internal shutdown switch. Once G-alpha is carrying GDP again, it loses its affinity for the effector and rebinds to the G-beta-gamma complex, reforming the inactive trimer. That's step 6.
This self-limiting design, this intrinsic timer, is probably the most fundamental safeguard against cellular chaos. The signal can't run forever without fresh input.
And to categorize the different jobs these timers do, we talk about 4 main flavors of G proteins based on their G-alpha subunit. There's GS, GI, GQ, and G1213.
Right. For example, GS is stimulatory. It activates an enzyme called adenylyl cyclase. GI is inhibitory. It inhibits that same enzyme. And GQ activates a totally different pathway, the phospholipase C beta pathway, which we'll get to.
Okay, so the cell needs backup mechanisms to prevent chronic signaling, a process called desensitization.
Exactly. It's not enough to just wait for the G protein to switch itself off. Desensitization happens on the receptor itself. This is steps 7 and 8 in figure 15.6.
So first, the activated GPCR gets recognized and phosphorylated by a specific kinase, a GRK, on its cytoplasmic domain. That's step 7.
Second, those new phosphates create a binding site for proteins called arrestins. That's step 8. And arrestin physically binds to the receptor and just blocks it, preventing it from activating any more G proteins.
This is so vital for things like sensory adaptation. We know this because mutations that mess with rhodopsin phosphorylation by its GRK lead to photoreceptor cell death.
Right, which causes the blindness seen in retinitis pigmentosa. If your visual system can't turn itself off, the cells just burn out.
And this arrestin protein is also the hub that kicks off receptor internalization.
Yep, it's shown in figure 15.7. Arrestins help link the receptor into these clathrin-coated pits, which promotes endocytosis, pulling the receptor inside the cell.
Once it's inside, the receptor has 3 possible fates. Fate 1, it can stay active in what are called signaling endosomes.
Right, so the arrestin acts as a scaffold to assemble new signaling complexes, which really challenges the old idea that all the signaling only happens at the plasma membrane.
Fate 2, it can get sent to the lysosome for degradation. That just temporarily reduces the total number of receptors on the surface, leading to a longer-term desensitization.
And fate 3 is resensitization. The receptor gets dephosphorylated and recycled back to the plasma membrane, getting the cell ready to respond to the next signal.
And to speed the whole system reset up, the G protein's own timer is often accelerated by accessory proteins called RGSs.
Regulators of G protein signaling. They just make sure the response is terminated rapidly.
The clinical relevance here is just immense. I mean, the chapter says that over a third of all prescription drugs target this GPCR superfamily.
And when this signaling goes wrong, the consequences are severe. A major source of disease is what we call gain-of-function mutations that make receptors constitutively active.
Meaning they're always on, even with no ligand.
Exactly. A clear example is mutations in the TSH receptor, which can lead to thyroid adenomas. The mutant receptor just continuously activates its G protein, which leads to nonstop SAMP production, excessive thyroid hormone, and uncontrolled cell growth.
And it's often a somatic mutation, one that just arises in a single cell, which shows how critical this tight regulation is for preventing cancer.
There's this fascinating rare case of a single G-alpha mutation that causes both precocious puberty and hypoparathyroidism.
And it's temperature sensitive.
Totally. In the cooler environment of the testes, the mutant G protein is constitutively active, triggering premature testosterone. But in the warmer parathyroid glands, that same mutant protein is inactive, which prevents parathyroid hormone secretion. A stunning example.
And of course, pathogens are signaling experts too. Think about cholera toxin.
It chemically modifies the G-AXA subunits in your intestinal cells, and it permanently inhibits their GTPase activity.
So it breaks off switch. The G protein gets locked on, continuously stimulating adenylyl cyclase.
Which results in this massive continuous production of CAMP, driving catastrophic fluid secretion and the severe dehydration that defines cholera. It's a direct attack on the cell's core safety mechanism.
Okay, we have to pause and just reflect on the discovery of the first ever second messenger, CMP, back in the 50s by Earl Sutherland. This was a huge conceptual leap.
It really was. He was studying how hormones like glucagon made liver cells break down glycogen. And the breakthrough came when he used broken cell preparations.
So he separated the cell into a soluble cytoplasmic fraction and a particulate membrane fraction.
And he found that the enzyme that breaks down glycogen was in the soluble part, but the hormone response only happened if he included the membrane part.
So his experiment showed that the hormone hit the membrane, causing it to release something that then diffused into the soluble part and activated the enzyme.
And that diffusible substance was cyclic AMP. It allowed a single external message to rapidly coordinate a huge metabolic change across the entire cell.
Now let's explore the phosphoinositide-derived messengers. These are products of lipid metabolism, which proves the membrane isn't just a container, it's an active signaling platform.
We start with a minor phospholipid called phosphatidylinositol, or PI. This gets processed by lipid kinases that add phosphates to its inositol ring, creating various phosphoinositides.
Okay, so the key path, which is in figure 15.10, shows PI getting phosphorylated to PI45P2. This molecule can then be phosphorylated by an enzyme called PI3K, PI3 kinase.
Right, and that creates PI345P3, or just PIP3.
And why is PIP3 so important?
It's a membrane-bound second messenger. It forms a crucial docking site for proteins that have a specialized interaction domain called the pH domain.
So binding the pH domain to PIP3 yanks the protein out of the cytoplasm and sticks it right on the plasma membrane.
Exactly. It places it in direct contact with other membrane-localized partners. Location dictates activity.
We can even see this. The chapter has these images showing PIP3 specifically concentrated at the leading edge of a migrating cell.
Right, figure 15.9c. That localized production is literally telling the cell which way to move. It's directing its chemotaxis.
Now for the other path, what happens when a G-alpha-Q subunit gets activated?
That stimulates an enzyme called phospholipase E-beta, or PLC-beta.
And PLC-beta splits that membrane lipid PI45P2 into two completely different second messengers.
Correct. The first is diacylglycerol, or DG. It's a lipid, so it's embedded in the membrane. The second is inositol 1000-5-trisphosphate, or IP3, which is water-soluble and diffuses into the cytosol.
Let's follow DAG first. It stays in the membrane, and it recruits and activates Protein Kinase C, PKC.
And PKC is a serine ethionine kinase that's really involved in regulating growth. The link here is so profound. There are these plant compounds called 4-ball esters.
They structurally mimic DAG.
Exactly. So if you apply them to cells, you activate PKC, and the cells temporarily start behaving like malignant cells. Continuous inappropriate PKC activation can lead to cancer.
Okay, now IP3. It's water-soluble, so it diffuses through the cytosol and binds to the IP3 receptor on the smooth ER.
And the IP3 receptor is a calcium channel. So when IP3 binds, it opens the channel, triggering a release of stored calcium ions into the cytosol. And calcium is just this universal, incredibly versatile messenger.
What's so amazing is the complexity of the calcium signal itself. The book shows that in liver cells responding to a hormone, the calcium release isn't a steady stream, it's rhythmic, repetitive bursts.
Right, calcium oscillations from figure 15.11.
Mm-hmm.
And here's the information coding part. A higher hormone concentration doesn't make the spikes bigger, it makes them more frequent.
The cell is using the rhythm, the frequency, to encode information.
It's incredible, and it all comes back to specificity. There are 9 different epinephrine receptors, 15 for serotonin, dozens of G protein subunits. The sheer number of possible combinations ensure that even if the signal starts with the same ligand, the outcome is precise to that cell type.
Let's look at a classic, beautifully illustrated example of this. Glucose mobilization in the liver, driven by CAMP.
This is a perfect example of convergence. 2 different hormones, glucagon, which signals low blood sugar, and epinephrine, which signals stress, both converge on the exact same pathway.
So they both bind their respective GPCRs, which activate the G-alpha subunit. This activated subunit then converges on the same effector, adenylyl cyclase, causing a spike in CAMP.
And CAMP acts by binding to the regulatory subunits of inactive protein kinase A, or PKA. This causes the release of the active catalytic subunits. That's all in figure 15.14.
And active PKA is the metabolic switch. On one hand, it phosphorylates and inhibits glycogen synthase, shutting down glucose storage.
And on the other, it phosphorylates and activates phosphorylase kinase,
Which then activates glycogen phosphorylase, the enzyme that actually cleaves glucose units from glycogen.
This sequence is a perfect demonstration of signal amplification. One activated G protein makes hundreds of CAMP molecules. Each active PKA can activate many molecules of phosphorylase kinase, and so on. A faint whisper of hormone becomes a metabolic roar.
And the whole thing is reversed by protein phosphatase 1, which removes the phosphates, and phosphodiesterase, which destroys the CAMP.
And importantly, PKA isn't just in the cytoplasm. Some of it moves into the nucleus, where it phosphorylates a transcription factor called c-Arab.
And that phosphorylated c-Arab binds to specific DNA sequences called CREs, stimulating the transcription of genes needed for metabolism, like making new glucose.
This takes us back to specificity. How does PKA in a liver cell trigger glycogen breakdown, but in a thyroid cell it triggers hormone secretion?
And the answer is these structural proteins called AKFs, PKA-anchoring proteins.
These are non-enzymatic scaffolding proteins. They physically tether PKA next to specific substrates in distinct parts of the cell, the plasma membrane, the nucleus, wherever.
So they're like the cell's internal organizers. By physically sequestering the shared kinase, PKA, right next to the relevant targets for that cell, you ensure that a global rise in CAMP results in the precise, appropriate local response.
Okay, the GPCR story ends with a look at how these receptors govern 3 of our senses: vision, smell, and taste.
Vision is the most dramatic one. A photon hits rhodopsin. It activates the G protein-Transduction, which activates cGMP phosphodiesterase.
And that enzyme rapidly hydrolyzes cGMP. So the key mechanism here is excitation by removal.
Right. In the dark, high levels of cGMP keep certain sodium ion channels open. When the light hits and cGMP levels plummet, those channels close, which hyperpolarizes the cell and generates the nerve impulse.
Smell relies on hundreds of different odorant receptors, also GPCRs. Each olfactory neuron is super specialized, expressing only one type of receptor, making it sensitive to just one or a few related chemicals.
Activation leads to CAMP synthesis, which opens a CAMP-gated channel, depolarizes the cell, and sends the signal.
And the vast range of smells we perceive is just the unique combination of these specialized neurons firing at the same time.
Taste is simpler. Salty and sour use direct ion channels, but sweet, bitter, and umami all use GPCRs.
Bitter taste, which protects us from toxins, uses about 30 different T2R receptors, all coupled to the same G protein.
While sweet taste uses only one high-affinity receptor.
And the rich, complex experience we call flavor is only achieved when the signals from your taste receptors merge with the olfactory input coming up through the back of your throat.
Which is why if you pinch your nose, food just tastes bland.
Exactly. The complexity vanishes.
Okay, let's shift to the second major membrane signaling system, Protein Tyrosine Kinases, or TKs.
Right. These enzymes phosphorylate proteins only on tyrosine residues, and this whole system is really tightly associated with regulating proliferation, differentiation, and cell survival.
And because of that, unregulated TK activity is a hallmark of cancer.
We can categorize them into 2 groups: The RTKs, or receptor tyrosine kinases, which are the integral membrane proteins that act as receptors for growth factors, and then the non-receptor TKs, which are just floating in the cytoplasm.
The activation mechanism for RTKs is really elegant. Receptor dimerization, which you can see in figure 15.1 scene. The external ligand forces two inactive receptor monomers to come together.
And this can happen in two ways. There's ligand-mediated dimerization, where the ligand itself is bivalent and it physically bridges the two receptors.
Or there's receptor-mediated dimerization. Here, a monovalent ligand binds, but that causes a shape change that exposes a new dimerization interface on the receptor itself, allowing them to link up.
Once those two monomers are brought close together, the game-changing step occurs: trans-autophosphorylation.
The kinase domain of one receptor phosphorylates tyrosine residues on the other receptor, and vice versa.
And this does two critical things. First, it activates the kinase domain itself. And second, the newly created phosphotyrosine sites, the p-tyr sites, act as specific high-affinity docking sites for cytoplasmic signaling proteins.
So those p-tyr sites are like molecular handles. What specialized domains grab onto them?
The two best studied are the SH2 domain and the PTB domain. The SH2 domain is about 100 amino acids long and has a conserved pocket that specifically recognizes not just the phosphorylated tyrosine, but also the short sequence of amino acids right next to it.
And that sequence specificity is what ensures different RTKs can recruit completely different sets of signaling proteins.
And the PTB domain does a similar job, also binding to p-tyr sites.
So this recruitment links the RTK to the cell's interior, bringing in 4 functional classes of downstream proteins. That's in figure 15.19.
Class 1, adapter proteins, like GRB2. These are just simple linkers.
Yeah.
GRB2 has one SH2 domain to grab the receptor and 2 other domains to grab a signaling partner. Its whole job is just to bring a signaling molecule to the membrane.
Class 2, docking proteins, like the IRS proteins. These are amplifiers. The RTK phosphorylates the docking protein, which then provides multiple new tyrosine sites, turning one receptor activation into a huge multi-site docking platform.
Class 3, transcription factors, like the STAT family. These bind directly to the receptor, get phosphorylated, dimerize, and then go straight to the nucleus to regulate genes, particularly in the immune system.
And finally, Class 4, signaling enzymes. These are enzymes like kinases or phosphatases that have their own SH2 domains. They get activated just by being recruited to the RTK, bringing them closer to their substrates.
And just like with GPCRs, the response has to be terminated, often by internalization. A protein called CBL binds the activated receptor and adds a tag, a ubiquitin tag, which marks it for endocytosis and degradation.
Now, for the most famous downstream pathway initiated by RTKs, the Ras-MAP kinase cascade. This pathway controls proliferation, and it's intrinsically linked to cancer.
Ras is the central player. It's a small monomeric G protein, kind of like a mini version of G-alpha, anchored to the inner membrane.
And it functions as a classic molecular switch, cycling between active GTP-bound and inactive GTP-bound states.
And this is such a critical control point because activating mutations in RAS genes are found in roughly 30% of all human cancers.
Those mutations essentially break the GTP hydrolysis, the off switch, locking res in the on state, and just continuously telling the cell to divide.
The cycling of Ras is tightly regulated, as you can see in figure 15.21. GTPs, or GTPase activating proteins, speed up GTP hydrolysis, turning Ras off.
And conversely, GEFs, guanine nucleotide exchange factors, stimulate the GDP release and DTP binding, turning Ras on. The key GEF here is a protein called SOS.
So the MAP kinase cascade, shown in figure 15.22, is this 3-tiered kinase hierarchy that's conserved across all eukaryotes. This sequential activation provides amplification and tons of opportunity for regulation.
It all starts with RTK activation and the recruitment of that adapter, GRB2, which is already bound to the GEF, SOS.
Bringing SOS to the membrane activates RAS, and RAS-GTP then kicks off the kinase relay.
A RAS activates RAF. RAF then activates the MAPKK, which is MEK, and MEK activates the final kinase, the MEPK itself, which is ERK.
Once active ERK, or MAPK, moves into the nucleus, where it phosphorylates various transcription factors, stimulating the expression of genes required for cell proliferation, like cyclin D1. But even this proliferation signal has a built-in break. One of the genes that gets turned on actually encodes a MAPK phosphatase.
So it's a delayed negative feedback loop. The pathway produces its own off switch.
Exactly. But this brings us back to that specificity problem. With so many different isoforms of these kinases, how does the cell make sure that Ras activation leads to proliferation instead of, say, a stress response.
And the answer is, once again, scaffolding proteins.
Right, from figure 15.23. These scaffolds are structural proteins that physically bind the three components of the kinase cascade, the KKK, the KK, and the K, together in a specific physical orientation.
So the scaffold physically prevents them from interacting with the wrong partners in other pathways. It enforces specificity. The yeast example is perfect. The same MAPKK can participate in either the mating pathway or the osmoregulation pathway, and it depends entirely on which scaffold protein it's bound to.
Okay, our final major pathway uses the insulin receptor, which is a unique RTK that manages glucose metabolism. It's unique because it's already a stable dimer, even when it's inactive.
So insulin binding just causes the internal kinase domains to juxtapose, leading to the usual trans-autophosphorylation and activation.
But crucially, this receptor signals mostly indirectly. It relies heavily on these IRS docking proteins. The activator receptor phosphorylates tyrosines on the IRS molecules, which then serve as these big, robust docking platforms.
This docking strategy gives it immense versatility. One insulin signal can trigger multiple downstream events at once by recruiting different SH2-containing proteins.
The major downstream cascade is the PI3-KKB pathway. IRS recruits and activates PI3K, which generates that membrane messenger PIP3.
And PIP3 recruits the critical kinase, PKB, also known as AKT, to the membrane. Once there, it gets phosphorylated and activated.
And the termination here is critical. The lipid phosphatase PTNN removes the phosphate from PIP3, shutting the pathway down. PTN is one of the most important tumor suppressors we know of. If you lose it, you get uncontrolled PI3K signaling.
So what does active PKB do?
Two critical things for insulin. First, it triggers glucose transport. PKB initiates GLUT4 translocation, which you see in figure 15.26. It makes vesicles full of glucose transporters fuse with the plasma membrane, dramatically increasing glucose uptake.
And second, it promotes glycogen synthesis. PKB phosphorylates and inactivates an enzyme called GSK3. Since GSK3 normally inhibits glycogen synthase, inactivating GSK3 effectively activates glycogen synthase, promoting storage.
And the clinical link is obvious. Type 2 diabetes, chronic high-calorie intake leads to high insulin levels. Over time, the target cells develop insulin resistance. They just stop responding properly.
Which forces the pancreas to pump out even more insulin, leading to high blood glucose and eventual exhaustion of the beta cells.
Finally, the chapter connects this whole pathway to longevity. Studies across many organisms suggest that downregulating this pathway, specifically IGF-1 and insulin signaling, correlates with an increased lifespan.
This introduces a fascinating evolutionary trade-off. There's a central nutrient-sensing kinase in this pathway called MTOR.
Right. High MTR signaling prioritizes growth and reproduction when there's an abundance of nutrients. Inhibiting MTR, for example, with the drug rapamycin, basically mimics nutrient scarcity. It shifts the cell's resources toward maintenance and repair, which extends longevity.
So downregulating the signal of abundance is linked to health.
That seems to be the insight.
Okay, we're in the home stretch. Let's talk about calcium as an intracellular messenger.
Calcium is the cell's most strictly controlled messenger. Resting levels in the cytosol are kept extraordinarily low, about 10 to the -7 molar, thanks to powerful pumps.
So the concentration gradient between the outside of the cell and the inside is about 10,000-fold. This means opening just a few channels creates an explosive signal.
We already covered one release mechanism, the IP3 receptors on the ER. Others include voltage-gated channels on the plasma membrane.
And critically, the ryanodine receptors on the ER. These are often opened by a small influx of calcium itself, a process called calcium-induced calcium release.
So a little bit of calcium coming in triggers a massive internal dump.
The calcium signal is so complex in space and time. We see those rhythmic oscillations, but also these highly localized microscopic bursts in small regions of a neuron.
And even dramatic propagated waves, like the wave that spreads across a starfish egg when it gets fertilized. That's figure 15.29.
So how does the cell refill these internal stores?
It uses a brilliant feedback loop called store-operated calcium entry, or SOCE.
Okay.
When the ER calcium stores get low, a calcium-sensing protein in the ER membrane detects the drop. It then clusters and moves close to the plasma membrane, where it physically recruits and activates a calcium channel in the plasma membrane. This opens the door for external calcium to rush in and refill the ER.
And calcium acts through mediators, especially a protein called calmodulin.
Right. Calmodulin has 4 binding sites. When calcium rises, the binding changes calmodulin shape, exposing these hydrophobic surfaces that act like an activated key, letting it interact with and regulate all sorts of effectors.
This is so universal we even see parallels in plants with the regulation of guard cells and stomata.
Yep, the stress hormone abscisic acid triggers calcium influx into the guard cell. This rise in calcium ultimately causes the cell to lose turgor pressure, deflate, and close the stoma to prevent water loss.
So we've detailed all these individual pathways, but the reality is they form a dense, interconnected web.
Absolutely. The source defines 3 forms of this interplay: convergence, divergence, and crosstalk.
Convergence is when signals from totally unrelated receptors all activate a common effector. For instance, GPCRs, RTKs, and even integrins can all converge to activate Ras and signal through the MAP kinase pathway.
Divergence is what we saw with insulin. One ligand activates multiple downstream pathways at the same time.
And crosstalk is when one pathway interferes with another. Figure 15.35 has a classic example. A KMP signal through PKA can phosphorylate and inhibit RAF, the MAPKKK.
So the metabolic signal literally puts the brakes on the growth signal.
But they can also cooperate. Both PKA and an MAPK-activated kinase can phosphorylate CRB on the exact same site.
Which brings us back again to that persistent puzzle. With all this crosstalk, how does the cell maintain any specificity?
And the answers we found are, first, protein isoforms, specialized versions of the same enzyme.
But overwhelmingly, it's the role of those scaffolding proteins. They maintain specificity by imposing a strict local spatial organization on the components.
Okay, let's briefly touch on nitric oxide, or NO, as a unique gaseous messenger.
Right. NO is synthesized from arginine, and it acts as both an intercellular messenger and a second messenger. The key discovery showed that smooth muscle relaxation could only be triggered by acetylcholine if the endothelial cell layer was intact.
So the endothelial cell was releasing some kind of diffusible substance.
And that substance was NO. In the blood vessel, acetylcholine binds its receptor on the endothelial cell, raising calcium, which activates NS to produce NO.
And since NO is a gas, it just diffuses right across the membrane into the adjacent smooth muscle cell.
Inside the muscle cell, it stimulates an enzyme called soluble guanylyl cyclase, which produces the second messenger, cGMP.
And cGMP then activates a protein kinase, which leads to muscle relaxation and vasodilation.
The medical relevance is staggering. Nitroglycerin for angina works because it's metabolized into NO.
And the mechanism of Viagra is a perfect downstream example. It doesn't affect NO production. It inhibits the enzyme that destroys cGMP, the cGMP phosphodiesterase.
So by inhibiting that enzyme, Viagra just prolongs the life of the cGMP signal, promoting sustained muscle relaxation.
And now for the final, most dramatic signal. The signal to die, apoptosis.
We have to distinguish this orderly programmed cell death from messy, uncontrolled necrosis.
Apoptosis is clean. Cell shrinkage, blubbing, fragmentation, and rapid phagocytosis with no inflammation. Necrosis is trauma, swelling, rupture, leakage, and inflammation.
And apoptosis is essential for development, like carving out the shape of our fingers and for eliminating dangerous or damaged cells.
The central executioners are the caspases. These are proteases that are synthesized as inactive precursors. Once activated, they are a molecular wrecking crew.
They cleave protein kinases, disrupting survival signals. They cleave the lamins, causing the nuclear envelope to collapse. And most importantly, they cleave an inhibitor of an endonuclease called CAD, freeing CAD to systematically fragment all the DNA.
There are two major pathways to activate them. The first is the extrinsic pathway, which is triggered by external messengers like TNF.
TNF binds to a death receptor. This causes the receptor to recruit adapter proteins and 2 procaspase 8 molecules.
And this clustering forces the 2 procaspase 8s to autocleave each other, forming active caspase 8, the initiator caspase, which then kicks off the downstream executioners.
The second, more complex pathway is the intrinsic pathway, which is mediated by the mitochondria and triggered by internal stress like DNA damage.
This pathway is tightly regulated by the BCL2 family of proteins, which act as checks and balances. Some are anti-apoptotic, some are pro-apoptotic.
Internal stress leads to the activation of specific proteins that neutralize the anti-apoptotic members, which frees up the pro-apoptotic members, BAX and BAC.
And BAX and BAC then form channels in the outer mitochondrial membrane.
This causes the release of mitochondrial proteins into the cytosol, most famously cytochrome C.
The release of cytochrome C is the irreversible point of no return. In the cytosol, it assembles with other proteins to form the wheel-shaped apoptosis complex.
And this assembly activates procaspase 9, which becomes the initiator caspase for this pathway.
The final step is clearance. The dying cell flips a lipid, phosphatidylserine, to its outer surface. This is a universal "eat me" signal.
Macrophages immediately recognize it and engulf the apoptotic bodies, ensuring no toxic leakage and preventing inflammation. It's a remarkably clean process.
But the cell's decision is always a balance. Most cells that get a death signal also get opposing survival signals. The final choice, to live or die, is a complex integration of everything.
Right.
We have completed our deep dive into the cell's complex communication system. Let's just briefly recap the core takeaways.
First, the GPCRs are the largest receptor family. They rely on that G protein switch, our molecular timer, to generate second messengers like CAMP and IP3.
Second, RTKs are the domain of growth factors. They use tyrosine phosphorylation to create instant docking platforms, activating key cascades like the Ras-MAP PK pathway for proliferation and the PI3-KPKB pathway for metabolism and survival.
Third, signals are processed through this web of convergence, divergence, and crosstalk, with specificity maintained locally by those non-enzymatic scaffolding proteins.
And fourth, the system includes the ultimate control mechanism, apoptosis. This programmed destruction, orchestrated by caspases, can be triggered externally by death receptors or internally by mitochondrial stress.
And that internal mitochondrial trigger, cytochrome C, always brings us back to a final, profound question. Why would this ancient prokaryotic-derived protein, essential for the cell's powerhouse, evolve to become the irreversible signal for self-destruction in complex eukaryotes?
It suggests a deep evolutionary repurposing, that the very engine of life, the mitochondrion, carries within it the mechanism for controlled death.
It hints that maybe the signal for cellular identity and cooperation includes, inherently, the mechanism for self-sacrifice.
A powerful thought to end our journey on.
Indeed.
Thank you for joining us on this deep dive into the operating system of the cell. We look forward to dissecting the next big idea with you soon.